everyone. I met some of you the other day, but well, for the ones that didn't know me before, I'm Luis Zapata. I'm a PhD student from the lab of Stefan that you already know. Me and Hannah have been working on tumor heterogeneity and exome sequence analysis of cancer samples, basically, and for that we have been, well, I've been involved in this project for already 4 years or more or less. But before I start with this, we have to celebrate San Jordi and, well, a flower for everyone and also a book that might be good for you is to start with the bioinformatics for dummies. At least I think it's a good start for you if you've never been involved on the bioinformatics field. Actually, one of the person that wrote this book is one of the PIs here in the institute. It's Cedric Notre Dame. So, well, there is no. And the other one is also, like, I have met this other guy, Jean-Michel Cliverici. He's also a very cool guy. Well, but then you have this book if you want to go into more deep understanding, not really more deep understanding, just a little bit more of what bioinformatics can help you. Okay? And regarding to the topic, so I'm going to start with, like, the typical slide that I use for introducing in my talks. The first is to tell you, like, it's been known for already 40 years that tumors resemble an evolutionary population where, like, you have evolutionary forces acting over these cell populations, and these cells acquire mutations that may confer them a selective advantage over the rest of the cells. So that's why they will become proliferative, highly proliferative, and then will overtake the cancer cell population that you will have in a particular tumor, if it's solid in a region or if it's non-solid in a mix in the blood, okay? So this is what I'm showing here. This is a piece of a tumor, okay? And what they did is they took a piece on this, a geographical sample, how it's called, and a piece of each of these regions, and then they sequenced these regions, okay? So if you expect that the tumor is homogeneous, so it all have the same cells and the same mutations, you will expect that they all share the same mutation and they don't have any differences between them. But it's not like that, because what we know is, like, it looks like an evolutionary process with, like, an evolutionary tree where there is an initial mutation, okay, which is often called the driver mutation, okay, and then these pieces can acquire more mutations and then can acquire new properties, and they can become metastatic or they can become, like, avoiding different apoptosis, and then you have these hallmarks of cancers as well, that they can be attacking, these mutations can be affecting one of these hallmarks, okay? So the idea of here is that basically this is why we have recognized that tumors are heterogeneous. It's called tumor heterogeneity. And this is observed in solid tumors. Well, if you have any questions, you want to stop me, please feel free to do it. And then there is also the same problem in if you have a non-solid tumor, a blood tumor, for example, what you will have is you have the founder population of cells. So you have one cell population with a particular mutation, it starts to expand itself, then it starts to acquire new mutations, here in blue. Then this population now, it acquires an extra mutation again, this in yellow, and then so on, okay? This can continue endlessly until you die or until you cure the tumor or whatever, okay? But what is happening here is, like, these cell populations are coexisting to each other, and they are, like, competing each other for the resources, for the niche. And often what we will observe is that one of them is more predominant than the rest. So this means that the cell population that is more predominant than the rest is the one that have a higher fitness. In evolution, what is called higher fitness, okay? So the idea is also, if you think about it, how to identify these mutations, what you want to know is, like, you want to know the mutations that are specific they are shared by all the tumors, but by all the cell populations here, and the ones that are also private for the ones that are more advantageous for the tumor, okay? As an example here, for a real example, what happened in usual treatments of cancer, what you do is you apply a particular drug, and after a while, you see that this patient relapse, has the relapse, no? And this can be because initially you kill all the cancer cells, you think that you kill all the cancer cells, but there's a few ones that have are resistant to this particular drug, this chemical. And now, since you change the environment of the cells, you have this one that is more fit than the rest. So it will outgrow the rest and it will become a new population here. So this kind of heterogeneity that is happening in tumors is some of the things that have been studied in the previous year a lot, because it's one of the things that will help us to solve more and cure more cancers in the future. Okay, so this is as an introduction. And how do we do this? I mean, we do it by sequencing. We do it by exome sequencing, and as you've seen, well, other types of sequencing also can help. But in this example, what I will show you is like you have the blood, no? And this actually is the part of the hospital, I don't know, the clinicians there, or the technician on the hospital, that they have to divide this blood into the tumoral and the normal ones. Okay, so the tumoral ones are these ones, and the normal ones are this one. And then you sequence, next. And then you sequence this population of tumor ones versus the normal ones, and then you get reads as you observe these FASTQ files containing this information here. And then what you do is it's very similar to what you have been doing. You align all of these to the reference, and then from the reference you have to identify which ones have a particular mutation here, which is not present on the normal sample. Okay, so this is what is called the somatic mutation. Because you think that all the germline mutations or the predisposition mutation that you might inherit from your parents, they are going to be present in all your cells. Okay, in your germline cells and not only in the somatic cells. So from this information here, what we have is what we count the number of times that we observe this mutation, and then we transform this, what is called the allele frequency, into a cancer cell fraction. Okay, I think you're lost a little bit here, because once we identify the SNP, we also have the information on how we want to know how many cells have this SNP. Not only that the patient have the mutation, we only want to know how many cells within this patient have the mutation. Okay, because this will let us know how bad this mutation can be. Because if it's a passenger mutation, for example, a mutation that doesn't have anything to do with the cancer, what you will see is that cells will acquire randomly these mutations, but they will never be in all the cells. So they will always remain at a low fraction of the cells. What I'm talking is, this is all novel things that have been studied for the last 2 years, and we have been exploring on this idea, and we have been working on this idea for the previous years to improve this gene identification or mutation assessment of the impact of the mutation. Okay, so next. So did you jump something before, no? Okay, maybe, okay. So regarding to the same, I think you have seen this slide already, or no, the talks. It's like what you want to do as a goal is to get the germline genome from a patient, the cancer genome, align this, and then do this variant calling, which includes SNPs, indels, which are insertions and deletions. I think you also saw them in the class. The structural variants. This is the copy number variants, or rearrangement, translocations, this type of mutation events. We will not go into that, but this is what is being done currently, is trying to assess all type of mutations that you can have in a genome and compare the cancer versus the normal, and it's not only going to be about the SNPs. So far, we still are studying basically all the effect of the SNPs, and we still don't have a comprehensive catalog of cancer mutations. So, well, we will see this in the workshop that we're going to do today. Then what you want to do is basically the annotation controlling, visualization, and variant effect prediction. Basically, this is what you have been doing for the diseases. It's not very different from the tumor. The only difference is that you have now a paired sample, a germline and a normal tumor sample. Then you obviously will have this, no? The tumor-specific findings, which are the somatic mutation-specific, or the cancer-related germline findings, so which these ones are going to be predisposition or inherited mutations, no? Then, next one, yeah. Next, what we're gonna do in the workshop today is to work in a CLL case, on a chronic lymphocytic leukemia case. The chronic lymphocytic leukemia is one of the leukemias that are around, well, the cancers, you know. Can you click? So basically, the lymphocytic leukemia come from the metapoiesis root, you know, from the B lymphocyte, well, the lymphoid cells, but specifically what we're studying here is the B cell chronic lymphocytic leukemia. So it happens here, when the cell acquires this mutation and it becomes cancerous, it becomes highly proliferative. You also have other ones in the myeloid, because some of you have worked on this more. So what we know from chronic lymphocytic leukemia, I'm not an expert, I'm not a clinician, okay? So I'm not an expert on the biological part of the leukemias. But what is known is it's one of the most common adult leukemias. You can observe an accumulation of this small B lymphocyte in different parts of the body. It's one of the most heterogeneous cancers as well. So the most common mutation, the most frequent mutation in leukemia is around 18%. So only 18%, between 15% and 20% of patients have a mutation in leukemia. So it's still a lot to explain. There is still a lot of patients that have no mutation linked. There's also two molecular subtypes that are very different from each other. One is resembles more to a stem cell-like phenotype, and the other one is not like a stem cell. It's more specific, more differentiated state. So this more stem cell-like is more aggressive than the non-stem cell-like, and the idea will be also to try to identify this genetically by exome sequencing, to say you have the highly mutated IgBH gene, which is called the IgBH mutated status, or the non-highly one, to see if there is a difference on survival or the prognosis for the patient. No? Next. So where do we get all this data from in our lab? We have the International Cancer Genome Consortium, okay, which is a catalog. We're part of that of this, and there is a catalog of around 500 CLL cases, and we have sequenced, well, not we, the people working on this have sequenced already 150 whole genomes, 500 exomes, 150 RNA-seq, and from all of this information, they're going to publish soon a paper which is trying to do this final summary of all the things that have been identified. Okay, but you will only do one case, and then tomorrow we will probably try to do a cohort of many cases, and you will see how it looks like, the files, and how you can identify the same thing that they did here in the big paper. Okay.